This video covers Concepts 3.1 and 3.2 of Chapter 3. An atom's electron configuration determines the kinds and numbers of bonds the atom will form with other atoms. This is the source of carbon's versatility. With 4 valence electrons, carbon can form 4 covalent bonds with a variety of atoms. This ability makes large, complex molecules possible. In molecules with multiple carbons, each carbon bonded to 4 other atoms has a tetrahedral shape. However, when 2 carbon atoms are joined by a double bond, the atoms joined to the carbons are in the same plane as the carbons. When a carbon atom forms 4 single covalent bonds, the bonds angle towards the corners of an imaginary tetrahedron. When 2 carbon atoms are joined by a double bond, the atoms joined to those carbons are in the same plane as the carbons. The electron configuration of carbon gives it its covalent compatibility with many different elements. The valence of carbon and its most frequent partners, hydrogen, oxygen, and nitrogen, are the building code that governs the architecture of living molecules. Carbon chains form the skeletons of most organic molecules. Carbon chains can vary in length and shape, as shown here. There are several functional groups that are important in life, biology. And these are the components of organic molecules that are most commonly involved in chemical reactions. And what gives a molecule its unique properties is a number and arrangement of functional groups. In biochemistry or life chemistry, these are the most important functional groups. The hydroxyl group, OH, is present in alcohols like ethanol. The carbonyl carbon can either be in the ketone, such as acetone, or in aldehydes, as propanol. Carboxylic groups are acidic. So here's acetic acid. It's acidic because it can dissociate its hydrogen ion. Amine groups can ionize, and they accept the hydrogen ion, so they're basic, and they form a positive charge, NH3. Sulfhydryl groups we'll learn more about later. They can form covalent bonds with each other. And here's the amino acid cysteine showing the sulfhydryl group. Organic phosphates are negatively charged and they have high chemical potential energy, such as what we see here in glycerol phosphate. Methyl groups are nonpolar covalent, and you see them on a lot of different compounds, such as methyl cytosine. So chemical groups can contribute to the function of molecules by affecting molecules' shape. In this case, the female sex hormone estradiol has attached chemical groups that different from the male sex hormone. So you can see the shaded in blue, you got differences here. Hydroxyl and estradiol in the same position, you have a double bonded carbonyl group, double bonded oxygen, and a methyl group for testosterone. Sometimes you can have chemical groups also in the effect of molecular function by being directly involved in chemical reactions. And these chemical groups are known as functional groups. ATP is a very important organic phosphate containing molecule. It's the primary energy transferring molecule in the cell, or the energy currency. And it consists of an organic molecule, adenosine, attached to a string of phosphate groups. So adenosine covalently attached to 3 phosphate groups. Where 3 phosphate groups are present in a series as an ATP, one phosphate may split off as a result of reaction in water. So you lose a phosphate as inorganic phosphate. And breaking that bond releases energy. And this energy can be used to drive biological processes, chemical reactions. Anything that can be used by the cell. The next concept here is polymers and monomers. A polymer is a long molecule consisting of many similar building blocks. These small building block molecules are called monomers. Some molecules that serve as monomers also have other functions on their own. Cells make and break down polymers by the same process. A dehydration reaction occurs when 2 monomers bond together through the loss of a water molecule. So when you form polymers, you lose a water molecule to add that monomer to this polymer chain. When you break down or disassemble polymers, water is added back to the system. So this is called a hydrolysis reaction. And you break this bond and form a shorter polymer and the monomer. Polymers can be quite diverse in cells. You have different macromolecules, and they can vary between species. And you have an immense variety of polymers that can be built from a small set of monomers. And that's sort of the basis of biochemistry. And that ends this video.